Do we put plus? ATP? Or just put over there that's generating all of the energy, all right? This is an oxidation reduction reaction, right?
Sorry.
Glucose is oxidized to carbon dioxide and water. Oxygen picks up the hydrogens, it's reduced to water, okay? All right, that's our first step. Then take and draw your box. Okay. What's the name of the first box? No. Put your camera down. You can take a picture when we're done. Okay, glycolysis. And what does glycolysis mean?
Break down
Give sugar split. Exactly. And that's what's going to happen in glycolysis. Sugar, glucose, is going to be split in half to 2 pyruvic acids. And what's the second one? Krebs cycle. Okay, sometimes it's called citric acid. Or even TCA. Same thing, all right? Okay, TCA stands for tricarboxylic acid. Citric acid has 3 carbonyls. Okay? All right, what's next? Okay, now you can put ETC, you could put E-transport, you could even call it oxidative phosphorylation. I'll stop right there because I'll just stop. Okay, same thing, right? Okay, now, next thing I want you to put, where does glycolysis occur?
In the cytoplasm.
Cytoplasm. Okay, Krebs cycle and electron transport occur in the
Mitochondria.
Okay. This is the matrix. This is the actually. All right.
Do you want us to know inner or outer membrane, or
Just say membranes. Okay. You can show over here in the electron transport that it's in the inner membrane, but it's related to the space between the inner membrane and the outer membrane. Okay. Got it. Okay. Next thing. Aerobic or anaerobic?
Anaerobic.
Okay, anaerobic. Well, what does that mean?
without air?
Without oxygen. No oxygen. No air. All right. Now, what is this? Aerobic. Now, we're going to say that, and I'm going to say it kind of tongue-in-cheek, okay? Because when we do the Krebs cycle, you won't see any oxygen. But if there's no oxygen to handle those hydrogen ions that are coming over here, the Krebs cycle shuts down. Okay, so I just say it's aerobic, even though if you take biochemistry, they'll say, all right, don't let them.
For now,
Concept.
we need to know.
This is concept, okay? All right.
Your class and your exam.
Okay. Now, what I want you to know for each one, let's just do it step like we've been doing. What does glycolysis start with? Glucose. What does glycolysis end up with? Two pyruvic acids. I'll just put PYR, okay? PYR. So that's what it starts with, that's what it ends with. Okay? What does the Krebs cycle start with? Pyruvic acid. 2 pyruvic acids. Okay? Which go through a prep step, remember, to form 2 acetyl acetyl-CoAs and a CO2. Okay? Now, some books will say 4 stages. Some books will say glycolysis, prep step, Krebs cycle, electron transport. I just kind of keep it simple by putting them together. Okay, now, we're going to see 2 acetyl-CoAs, 2 pyruvic acids, 2 CO2s. So we're going to actually show 2 circles, right? Okay? So what starts the Krebs cycle? Pyruvic acid. What are the end products of the Krebs cycle? CO2. You get a CO2, so CO2 and NADH. How much CO2? 2. 1, 2, 3, 4, 5, 6. 6 CO2s. So all of the carbon dioxide comes from the Krebs. Okay? Glucose, completely oxidized to carbon dioxide and water, is converted to carbon dioxide. And the carbon dioxide all comes from those chemical reactions in the matrix which are associated with the Krebs cycle. Okay? And every time it produces a CO2, it rips off 2 hydrogens and electrons, which have to be picked up and carried by NAD, or a carrier like NAD. Okay, FAD works too. Okay? All right. Now, so the Krebs cycle starts with pyruvic acid, produces carbon dioxide and water. All right, what is the electron transport chain, which this is a piece of the inner membrane? And this will be our chin. Okay? What does it start with? NADH. What does it end with? H2O. All of the water comes from where? Electron transport oxidative. Wait, we should put ETC. Oxidative phosphorylation. Okay? Now we'll come back and do the details. Okay? All right? We're gonna fill in a couple of months here then. Okay? Got it? Okay? Now, let's go over here. Let's go back to glycolysis. Good place to start is at what do we do? All right? How many carbons is glucose? 6. How many carbons is pyruvic acid? 3. 2 times 3 is 6. Look at the formula. 6 carbons, 6 carbons. Matter and energy are never created nor destroyed. They're only changed in form. That's the first law of thermodynamics. Okay. Now, if you remember, there's an investment of 2 ATPs for every molecule of glucose. So they have to have a steady supply of ATP to even make glycolysis turn. Now, if they invest 2 ATPs in glycolysis, they've got to follow, you've got to make more than you invest, right?
Right.
And so what you get is you get 2 ATPs on this side.
Is it not ADP in the chart? You have ADP.
No, it should be, this is ADP, okay? Because the phosphate literally gets transferred to the glucose. Okay, I'll go over that in a second. Okay, so 2 ATPs are formed into 2 ADPs, the phosphates go straight on. It makes 2 ATPs, makes 2 ATPs. So what's this? You invest 2, you harvest 4, what's your net gain in glycolysis? 2, that's all. Now, if you want to go into the details so you understand it a little bit better, just look at the picture in your book, okay? It shows there's an enzyme that takes glucose and ATP, rips the phosphate off ATP and glues it to glucose to form glucose phosphate. Glucose phosphate is not glucose. It's not glucose phosphate. There's another enzyme waiting for glucose phosphate, and it changes glucose phosphate to fructose phosphate. Fructose phosphate's not glucose phosphate. It's changed. It's different. Okay? Glucose phosphate, there's an enzyme waiting for it that grabs hold of it and another ATP and puts another phosphate on the other side. Now it's fructose 1,6 diphosphate or fructose diphosphate. That's your investment steps. In other words, they've got to prime the pump to get any energy out of it, and they get more than they get out. If you've ever worked on an old ranch where they didn't have an electric pump to the well, right? You had a well that you had to pump. Usually sitting right next to the pump was a bottle of water. And you had to pour the water in the top of the pump, and then all of a sudden it would pump, and you pump your water out, and you had to fill your jug back up for the next person, right? Because sometimes you have to invest energy to get more energy out. Okay, anyway, so what I want you to see in glycolysis that occurs in the cytoplasm that's anaerobic, you start with glucose, end up with 2 pyruvic acids, invest 2 ATPs, harvest 4, net gain of 2. Now, you have to have a carrier molecule. Carrier glycolysis is NAD. And since it splits into 2, you need 2 of them. Okay? And the NAD, 2 NADs will pick up a hydrogen and electron. Each will be converted to NADH. Okay? Because you can't let it. If you let a hydrogen ion just float off into the cytoplasm, what does it do to the pH? Makes the pH increase the acidity. The pH drops like a rock. Kill it. And so, and anyway, the enzymes that do this have to have a coenzyme, NAD, to work. An enzyme and a coenzyme go together. All right, so much for that theory there. All right, now, once we've got the NADH formed, in aerobic metabolism, like what you do in your cells, just as fast as the pyruvic acid is formed, it goes here, right? And as fast as this NADH is formed, it goes here, if there's oxygen. That's what we do, okay? But what do microbes do? Once we've been working on in lab, they're fermenters. They're either anaerobic, facultative anaerobes, or aerobes deprived of oxygen. What do they have to do to pyruvic acid? What's the term? You should use the word. What's the term? You have to regenerate the NAD from NADH, and the chemical process is called fermentation. So, all right, fermentation. That's what we're doing. We do our MRVP test. That's what we're looking for. Did the bacteria ferment glucose? And the way we test it to see if it's acid. All right? Now, fermentation is a set of reactions where the pyruvic acid has the hydrogen and electron glued back on it. So it's no longer pyruvic acid, it's now lactic acid. And that's what our muscles do when we're deprived of oxygen, or we don't have enough oxygen. Now, there's tons of different products of fermentation. Okay? But the most common are lactic acid, alcohol, and carbon dioxide gas. If you pour in a bubble like that, air like that. Now remember when we did our fermentation broths last time? Okay? We looked for yellow, that's acid. And if we saw bubbles in the tube, it's gas. So some bacteria ferment with acid, some with gas. Some with acid, or some with acid, some with acid and gas. All right. Now, there's tremendous varieties of these. And if you'll look in your lab manual, there's all these different fermentation pathways. When we do the BP test today, we're actually checking for a different end product that's not the same as lactic acid or acetic acid or whatever. There's all kinds of weird ones. And biochemists have been figuring this out a long time. That's why we have all those biochemical tests in Berkey's manual. Because those different end products can be used to detect what species you've got, so you know genetically which one it is. Okay, so think in your mind. What's the function of fermentation? To regenerate NAD. All right, in anaerobic glucose catabolism, glycolysis followed by fermentation, the final acceptor of electrons is an organic molecule. Lactic acid, acetic acid, all these are organic molecules. Because over here you're going to see the final end product is water because oxygen is grabbing it. Okay, all right. So in anaerobic metabolism, no oxygen. The final electron acceptor is an organic molecule. Now, how much ATP can you get through fermentation? 2. 1 microbe gives 3. But all right, mainly 2. All right.
Which microbe gets 3?
I don't know. We'd have to go back and look at it. Okay. All right. Now, we're done with glycolysis, right? Put a fork in it. Temperature's at 165. If it's a piece of turkey, we can eat it. It's safe. Okay? Now, glycolysis in aerobic organisms is followed by the Krebs cycle. Right? Now, if you're a bacteria, you don't have a mitochondria. So, the Krebs cycle enzymes are found in the cytoplasm. And the electron transport chain is in the inner membrane. Because it's got one membrane. That's all it's got. And so, a lot of the stuff that we know about this have come from studying bacteria. But now, in eukaryotes, they have this organelle that we call the mitochondria, that if we slice it like that, we can see it's got an outer membrane, OM, an IM. What's the IM? Inner membrane. And what's this here? What do we call the gooey center? Like in a Twinkie, the white stuff. The matrix, okay? That's the matrix. Okay, if you can think matrix, make yourself a couple of circles like that. What's going on in the matrix? Krebs cycle. And if you look at the IM, and if you just draw some lines over here, what's that? Electron transport chain. Now, you got to think with this. You got to kind of visualize in your mind